Good morning, everyone. If there is any difficulty hearing me or seeing the slides, please let somebody know through the system. But assuming that everybody can hear me and see the slides, I'm going to start our presentation. And I want to start by just talking about how exciting it is to be about to launch our COVID-19 vaccine program here at Jacksonville Center for Clinical Research. And the purpose of these educational talks, and this one is specifically geared towards physicians, is to help our community understand what reasonable expectations are as we move forward with these vaccine studies. Obviously, this is in the news all the time. There's a lot of information that's either not perfectly clear or slightly misleading or colored by political orientation and not necessarily as scientific as it should be. So our goal here at Jacksonville Center for Clinical Research and throughout the Encore Research Network is really to provide reliable information for our community, both physician community and patient community. And this program is one of those efforts. So probably the best description of a virus that I've read is the virus is simply a piece of bad news wrapped up in a protein. And as most physicians understand, viruses are very simple organisms, barely even alive, that don't even have enough genetic makeup in order to run their day-to-day functions. In fact, they have to rely on other cells in order to provide a lot of the services that are required to run a cell. So basically, a virus is a bunch of genetic material that takes over a cell and then reproduces itself. With that said, viruses cannot really live without other cells. So one of the misconceptions that you hear out there is that a cell virus is going to sit on a countertop and live for an extended period of time. Well, that actually doesn't happen. Viruses need cells in order to live and to reproduce. Their ability to survive without cells is literally measured in minutes. The other thing that is not well known, but I think is important, is that there has been a long history of making vaccines, and that history is actually being changed as we speak. So the vaccines that we're looking at for COVID-19 are actually quite a bit different than traditional vaccines. So I'm going to spend a little bit of time talking about how vaccines are traditionally made, but also remind this physician audience that the concept of vaccination is actually an ancient practice. And it goes back probably 400 or 500 years. And vaccinations were considered folk remedies, where in the case of smallpox, people would take some of the material from the pus of somebody infected with smallpox and actually inject it into a person who was not ill. And what they found is that if you injected it in the skin and the leg, for example, people would develop a rash from that, but they actually wouldn't get sick in the same way as if you got infected through the respiratory route. So the way that you actually were exposed to a virus had a lot to do with how sick you got and what type of course you'd have. The other thing that people learned was that once people were exposed to viruses, they tend to be immune. So back in the smallpox days, that was a very interesting understanding, and the first modern clinical trial actually happened in the 1720s. And it was at the notorious Newgate prison in Great Britain. And there was a study that was done in 7 people there who were exposed to smallpox, and all 7 agreed to participate in the study in exchange for being released from prison, which obviously would be unethical by today's standards. But at that time, 7 people came in, and they had a surgeon make a cut in their legs, and the pus from smallpox was put into that wound. And 6 of the 7 actually developed a reaction to this procedure, and the 7th didn't, and I'll get to that in a second. But all the people that participated in the experiment were free from smallpox during a very intense epidemic that occurred in Great Britain of smallpox at that time. The other thing that we learned from early studies is that there is cross-reactivity and cross-immunization. So for example, ultimately the vaccine for smallpox, which has now been eradicated, actually came from cowpox, which was a similar but less virulent viral infection. So this concept of cross-reactivity is a very important one, and vaccines can be effective without being exact duplications of the virus that you're trying to protect people from. Getting back to the Newgate prison story, the 7th of the people that were involved wasn't able to mount any kind of rash or significant reaction to the initial surgery. And it was a woman that went through a second surgery in order to prove that she was actually exposed adequately to the smallpox in her leg. And after a huge gash that was put into her leg with lots of pus, she eventually had a little bit of reaction. And what turned out to happen was that when they studied this woman a little bit more, she finally admitted under intense questioning that in fact she had had smallpox earlier in her life, but that she lied about it in order to get into the clinical trial and to get out of prison. So that's actually a cautionary word for us in the research community. You have to talk to your patients and you have to ask them, and not always assume that everything that you're told is true and to be accurate. So getting back to how vaccines are made, over the last 100 years, the most common course of making a vaccine actually required growing the virus. Typically, these were in chicken eggs. And there were some ways where we learned over the last 10 or 20 years that we can improve this process. When you grow viruses in chicken eggs, it tends to be a relatively slow process. Plus, you have the challenge of having other antigens contaminate the ultimate preparation that could cause allergic reactions. So you've probably heard the problem in some patients who develop reactions to eggs. People with egg allergies may have a reaction to certain of the vaccines that are available. So because of this, alternative ways of growing viruses have been studied. And here at our center, for example, we've done studies that have looked at growing flu viruses in tobacco leaves. And it turns out that tobacco leaves are a very effective way to grow flu viruses. And they're actually a way of developing vaccines more quickly because the virus grows faster in tobacco leaves than in egg media. And then it's much easier to isolate the appropriate antigens from vaccines that are made in tobacco leaves as compared with those made in an egg-based preparation. Once you grow the virus, then you have to identify the antigen that you want to use to elicit the immune response. And in this case, antigen is just a protein that the immune system will recognize. And in some cases, the antigen can be the entire virus or a big portion of the virus that is inactivated. Obviously, it's very, very important through this process that we don't inadvertently give people active virus that can cause illness. Once you grow the virus and identify which antigen you're looking at, then there's a very extensive process to make sure that you purify that antigen and that ultimately the vaccine contains only the antigen of interest and not other contaminants. And as I mentioned, making sure that there is no inadvertent active virus in your preparation is extraordinarily important. And a lot of pains go through this process of making sure that there aren't very, very low levels of active virus when the vaccine is being made. Finally, after you identify the antigen that will elicit the immune response, you need to add other materials that will hopefully present the antigen in a way where our immune systems can recognize it. And also perhaps use stabilizers to prolong the shelf life, which can involve preservatives. And sometimes these preservatives actually become something that patients can be allergic to. So I'm sure as physicians, you've heard people say that I can't take this, that, or the other vaccine because of an allergy to preservatives. And there is some truth to that and some cautionary tales and some learning that's involved with that understanding. Once the vaccines are manufactured, then there's another scale up to make them manufactured in large quantities. They have to be put in vials, and then they have to be transported to various clinics and places that will be used around the world. Now, the vaccines that we're looking at now are using an entirely different strategy of creating immunity. And this is really important for us in the physician community to completely understand and to address these new technologies with our patients. So the two studies that we're about to get started here at Jacksonville Center for Clinical Research and the OnCAR Research Group are actually using what's called the mRNA platform. There are two different companies that are using slightly different concepts, but the bottom line is that we're not injecting viruses, we're actually injecting messenger RNA. And the theory here is that instead of giving patients some portion of the virus, what we're doing is we're teaching our own bodies to produce a key antigen or a key protein that's specific to the virus. And our body's smart enough to know what's a viral protein and what's a human protein. And once we express this protein, our bodies will identify it as a possible infection and create an immune response without actually having the virus in our system. So these are very important concepts, is that these are just proteins that can infect anything because they're relatively simple and only one antigen of an entire virus. And two, the viruses are never actually touching our body. So when you think about it, we're not actually giving an inactive virus, we're just giving a set of instructions so our immune system starts its natural process of creating antibodies against a protein that shouldn't be in our body. So just to refresh you from high school biology, messenger RNA is just a set of instructions. As you recall, DNA is the genetic code, and then we create RNA, which is a way of taking the information from DNA and turning it into action. And messenger RNA goes through the process of translation in a ribosome, and the ribosome in turn creates the protein. So, as I mentioned, we are working on 2 vaccines, but there's a lot of work in vaccines around the world. In fact, there are already 145 COVID-19 vaccines in development using different methodologies. Some of the old traditional methodologies using eggs, using other media to grow the viruses, and others are using these genetic platforms. And the ones that we've been interested in, the ones that we've been awarded, have involved these genetic platforms. So far, there have actually been 16 vaccines that have been manufactured and tested in humans. So of the 145, over 10% have already touched human beings. And the vaccines that we're working with have also been tested in humans in phase 1. We can talk a little bit about that towards the end of the talk. There has never been so much intellectual firepower put into a vaccine program in the history of medicine. So I think you're going to be hearing a lot of information about this in the media and through talks such as this webinar, where we can update our community and make sure that people are really understanding what's truly happening versus the hype and the media spin. But already phase 1 results have been released, both from the Moderna program and from the Pfizer program, and both have shown positive results of being able to create antibodies against key components of COVID-19. Obviously, with all this work going around the world, no one knows for sure which vaccine will emerge as the best. I suspect there will be more than one that emerges, but the mRNA platform vaccines actually are out in the lead for a number of reasons. One is that they can be produced more quickly, and two, they are cleaner from a theoretical standpoint in that you're just producing very, very specific proteins that don't have to go through an extensive purification concept in cell cultures, as I showed you previously. So this slide shows you the typical time frame between identifying a virus and then ultimately creating a vaccine. So if you look at the bottom of the slide, you see that for the H5N1 avian bird flu, which was first identified in 1996, it took over a decade before there was a meaningful vaccine. But that was useful because if you look up and you see the H1N1 swine flu, we actually had a vaccine on the shelf when the H1N1 swine flu was identified. So just to make that clear for everybody, remember, influenza is a series of viruses. It's not one virus. And you identify which type within the series based on this nomenclature of H and N1, which are 2 of the 8 proteins on influenza, which is an RNA virus. So the H5N1 was one type of influenza virus, and the H1N1 was another type. But because the vaccine work that had already occurred, we had an H1N1 vaccine on the shelf, and we were able to deploy that relatively quickly once we identified the virus. SARS is is a chronotype virus, and a very virulent virus. And you can see that there was no vaccine developed, but the good news is that because of the extreme virulence of it, it was identified quickly and isolated so that it wasn't widespread. A similar thing happened for MERS as you look up the slide, and you can see that that was active in different parts of the world between 2012 and 2015, and there was no vaccine candidate that ever became commercialized, largely because we were able to control it through epidemiological means. Moving up the slide, you can see Ebola, which is an absolute devastating viral illness, and it's one of these viral illnesses that can kill up to 90% of infected people. And we do have a vaccine now available for that, but it took about 5 years. And COVID-19, which was recently identified, as you know, in late 2019 and early 2020, is now in the process of having these vaccines developed at a pace that is completely unprecedented. So this slide is from the New England Journal from a couple of months ago, talking about how vaccines are usually made and how this COVID-19 vaccine is anticipated. So if you look in the top of the slide, you can see that vaccines tend to have a period of development of 5 years. And you can see from preclinical trials to phase 1 to phase 2 and phase 3 to licensing and large-scale manufacturing, it's an extensive process. But for COVID-19, we are going to compress this so that we anticipate having a vaccine available within 18 months. Now, again, because of the change in technology, some of the things that we had to do in preclinical trials and in small-scale production are not necessary for COVID-19. And this is one of the major advantages anticipated for the mRNA platform. So just to remind everybody, there's basically 2 ways to become immune to viruses. And I think it's important to explain this to patients because there are gaps out there. So there's really only 2 ways. Either you get sick, you get the disease, you recover, and your body learns how to mount an immune response that involves both antibodies and cell-mediated immunity. So for example, you get the measles, and once you get the measles, you have the memory in immune cells to keep you immune for the rest of your life. And then the other way to become immune to viruses is to not get the disease, but get a vaccine, which trains your immune system on how to recognize the virus so that when you are exposed to the virus, your body will kill the virus and protect you from the disease. So looking at SARS-CoV-2 or COVID-19, you can see in this slide that in the blue, that after becoming exposed to a virus, the viral titers grow very, very rapidly. And somewhere usually between 5 and 7 days, your immune system starts to kick in. It revs up, it starts to recognize the virus and create the ability to counteract the virus by targeting specific proteins in the virus. And the first immunoglobulin, I should say the first antibody that becomes produced is called IgM, which is a very, very large molecule, but is also the start of the immune process. And IgM in turn creates IgG, which typically becomes evident in our sera about 14 days after infection. And then IgG revs up to a very, very high level, and IgG becomes the primary immune protection for our immune systems, which helps deal with the viruses both by targeting the viruses directly, but also activating cell-mediated immunity, where we have cells that are going to take viral particles and actually pull them out of circulation, or take cells that have been infected by viral particles and take them out of the circulation. So this slide is a good way to understand the time frame between infection and the activation of our immune system. Again, IgM being the first step, and then IgG being the more robust second step that takes a little bit of time. So just to remind you again, going back to high school or early college biology, there are 5 types of antibodies. IgM and IgG are 2 of the 5 types. IgM has actually 5 subunits that you're seeing here on the left of the slide. So it's quite a large molecule. And you also, this slide also points out the FAB, which is the part of the antibody that actually binds the antigen. And then in the yellow, you see the part of the antibody that's just structural components of the antibody that allow it to circulate and to seek out its target. The other thing I'm going to be talking about is this concept of binding versus neutralizing antibodies. And it's important to know that not all antibodies that are produced will necessarily neutralize the virus or neutralize the key protein that we think is pathogenic. And this distinction is extremely important and part of what we'll be studying in clinical trials. So just by way of definition, a binding antibody can be produced at very high levels, and that's just a response to the overall virus and the overall foreign proteins, but they may or may not produce immunity. Whereas a neutralizing antibody is very specific to the target site that will either allow the virus to infect a human cell or be part of the pathogenesis of the virus. And I'll show you another slide that'll help you understand that. So this is important when you think about areas in which we've been able to use vaccines to produce positive medical results versus areas where we've used antivirals. So for example, hepatitis B is a disease where we've basically reduced its incidence to a huge degree using a vaccination strategy. So all healthcare workers and anybody else who's high risk has, I'm sure, been vaccinated with hepatitis B. And what was once a scourge is actually relatively rare. Hepatitis C, on the other hand, has been basically cured using antiviral drugs. And the difference between hepatitis C and hepatitis B is that hepatitis C is able to reproduce itself without being exposed to the circulation. So there can be a cell-to-cell reproduction of hepatitis C that doesn't involve release to the circulation. HIV, as you know, is also another virus where we don't have a vaccine, but that has been neutralized using antiretroviral type of drugs. And HIV has been difficult to treat with a vaccine because of the fact that it actually hides from the immune system. It hides in T cells and doesn't get represented to the immune system like other viruses. So this is important to understand, is that we're not sure if COVID-19 will be more like the flu as a similar type of RNA virus, or more like these other viruses, although we expect it to be more like the flu. And viruses have mechanisms where they can either hide from the immune system or evade it. And if that's the case, then perhaps an antiviral drug strategy will be better than a vaccine strategy. Obviously, the nice part of a vaccine strategy is you actually prevent infection, whereas when you use a drug strategy, you're treating people who are infected. So one of the questions that has come up is about coronaviruses, and coronaviruses in general, and specifically have abilities to mutate, and whether or not that will offset the effects of our vaccines. And of course, the people who are developing these vaccines are aware of those characteristics and are taking great care to target specific proteins that are, one, less likely to mutate, and two, are very specifically involved in the pathogenesis. So just to take a step back, just remind you that the term coronavirus is similar to the term flu virus, meaning that there are a lot of different coronaviruses as there are a lot of different flu viruses. Coronaviruses are typically in the animal kingdom, and they don't necessarily transfer over to humans very often, but they can mutate to forms that allow them to infect humans. So we know, for example, for COVID-19, that a mutation occurred that allowed the spike protein, which is the characteristic protein of coronaviruses, to connect and able to invade human cells through the ACE2 receptors, particularly ACE2 receptors in the lungs and in tissues in the respiratory tract. So because of that knowledge, the vaccines that we're developing are specifically targeted to these spike proteins that have affinity to the ACE2 receptor, with the concept that, okay, well, if that protein does mutate, well, chances are it's going to mutate to something that won't be able to infect our cells, because it has to be specific for that ACE2 receptor in order to infect humans. So I think there's a nice little picture, courtesy of our friends at Amgen, that give you a sense for what the difference between a binding antibody and a neutralizing antibody. So on the left side of the slide, you can see what they call a non-neutralizing or binding antibody. And in this particular case, the green structure, I get a little house there, a rectangle with a triangle on top of it, is the virus. And you can see the typical form of the antibody, which is attached to the virus, but not attached to the virus at a place in which it binds to the cell. So that would be considered a binding antibody, and something that may not be effective. Now, keep in mind that if the antibody is a large molecule, it may still get in the way of binding. As you can see, that's going to flip around and maybe have some effects on the conformational structure of the virus, which would make it more difficult for it to actually hit the receptor in a human cell. But nonetheless, we can be assured that that type of antibody is going to prevent the virus from infecting cells. On the right side of the slide, however, you can see that the antibody is very specific to the part of the virus that will actually attach to the human receptor. And once that occurs, the virus is neutralized. It can't infect the human cell under those circumstances. So what we know about neutralizing antibodies for coronaviruses is that patients who recover from actual infections mount very high titers of these neutralizing antibodies. And different people differ in the amount of antibody that they make. So that's also something that we're learning, and maybe having something to do with the differential vulnerability of different patient populations. But we don't know how long this protection will be in place. So do these neutralizing antibodies create a memory so that people can never be infected by this type of coronavirus again? We don't know that. And how long do these neutralizing antibodies circulate, and at what levels do they need to circulate in order to prevent recurrent infections, or in the case of vaccine, to prevent a primary infection? But what I will say is that never in the history of medicine has there been this much intellectual firepower and these many resources focused on a specific disease. So I have very, very high confidence that the programs that are being launched now will lead to success in the relatively near future. Again, we can't guarantee the results of any individual program, but I think the messenger RNA platform and the 2 studies that we're working on in this platform have really very promising initial results, and I think a reasonable level to be something that will help our patient population. But again, this remains to be seen through the vigors of the clinical trial process. So I'm going to stop there and address questions. And the last thing I want to have people understand is that when you do clinical trials, people sometimes might have the misperception that everybody in the trial will get the vaccine and they'll be protected. So it's very important that as a physician community, we explain to people that these are placebo-controlled trials. Not everybody will get the vaccine, but they will be part of a process and be ambassadors of knowledge. So both the physician groups that get involved with these studies and the patients who are involved in these studies are very important ambassadors of knowledge that will help us solve this really a huge public health crisis. So that's a term we like to help people identify with, rather than thinking that the product itself is going to be the wonder drug, because we just don't know that. We have high hopes, but we can certainly assure people that they will be ambassadors of knowledge and help solve this issue. But the individual product may or may not be the one that is ultimately successful. So I think we had some questions. I'm going to pull up the chat box. Yeah, so a lot of the questions that we've been getting from our patient population are things that I think most physicians intrinsically understand, but I think it's also important just to be clear about it. So one of the questions we get is, will I be injected with a live virus? And the answer to that is no. And for the mRNA platform, it's not even close, because we're just giving a genetic instruction. So we're not even giving the real proteins from a virus. We're just giving the instructions for our own bodies to make it. So that should be the purest way of presenting a viral protein to our immune system. So we want to be clear about that. The other thing that we've been asked is about whether or not this involves cell cultures with embryos or other things. And again, for the studies that we're doing with the mRNA platform, the answer to that is no. And let me read some of these other things here. The question is, when are we going to start? We are actually really excited about the fact that we are anticipating giving our first doses of vaccine and research within the next 10 days here in Jacksonville. So we're super excited about that, and we'll be one of the first locations outside of the phase 1 units to be able to provide the vaccine for community. Let's see. A number of my patients are concerned that they will contract the virus if they receive the vaccine. What's the best way to address the concerns? Again, reassuring them that they're just getting a set of instructions for their own immune system. They're not getting the virus. These sets of instructions don't even come from the virus except through knowledge-based systems. So it's not as if we're taking the virus and growing it and taking a piece of it. We're actually, because we've been able to look at we've been able to identify and discover the genetic code of COVID-19, all we have to do is create the set of instructions in messenger RNA. So we're just taking the sequence of very specific proteins and taking that sequence and creating a messenger RNA molecule, putting it in a little lipid particle so that we can inject it into the circulation and just telling the body what to do. So the whole technology is just truly remarkable, and you can just help people understand this by saying is that there's no part of a real virus that's ever going to touch them. This is just a little piece of genetic code that they'll be getting as part of this vaccine. I think there were a couple other questions.
address the Latinos and Blacks? Okay.
Yeah. So one of the things that is just a reality in our current environment is that there's a lot of sensitivity around ethnic background and race. And there are some issues related to this. So, for example, if you look at the statistics for coronavirus infections, COVID-19 infections in a lot of communities, particularly in Florida, particularly in California, you're seeing that there's been a disproportional impact on the Hispanic community. And you don't hear that much about it in the media because of the sensitivity around identifying ethnic groups or racial groups. But for us in the physician community, I think it's important to know that there may be elements of Hispanic culture, Latina culture that will make the virus become more prevalent in those communities, whether it's the living conditions or the culture or working requirements, et cetera. But bottom line is, as physicians, we need to care for all of our patients, and we need to help those communities know that they may be particularly vulnerable and perhaps really good candidates for research moving forward, so that we can, one, let people know in those communities that the research process is a safe and comfortable process for them. And again, I say it's safe because of all the scrutiny that goes in. We are actually studying the safety when we do research, but we will do everything possible to make sure that people have as safe an experience in it as possible. And we know from a lot of literally just decades of work doing clinical trials that people that participate in clinical trials in that system tend to have better outcomes than people in just standard medical settings. In terms of the African American community, actually there is no increase in prevalence of infections in the African American community, but there is a lot of sensitivity around racial issues now. And historically, Blacks have been more skeptical about research, going back to the Tuskegee problems that we've had. And in my experience, the best way to approach certain African Americans who you perceive are having concerns about being involved in research is to just say that, hey, we know about some of the past misdeeds. We know Tuskegee was a horrible misuse of knowledge and just a tragedy. That actually was a study that was conducted by the US government between the 1930s and 1970s. But in the modern era, that could never happen, and we wouldn't let it happen. And again, as an investigator, and all the principal investigators should be very clear about this, is that my job is to make sure that my patients have a good experience. We don't have, we'd love to see this vaccine work, but at the end of the day, we don't have any vested interest in the commercial success of the vaccine. We are here to have our patients get the best possible experience, perhaps get some protection from this new technology, but most importantly, become an ambassador of knowledge. And certainly the African American community is one that may be more skeptical, but in my experience, through just being truthful about the history and be truthful about the current process, usually you can overcome some of those levels of suspicion. So now these are just general COVID questions now. Okay, so one of the issues that has come up about wearing masks. This has become something that has been politicized, unfortunately, and I'll give you my spin on that. So obviously there are different ways of transmitting COVID-19. Through respiratory droplets is certainly one of those ways. And what I like to remind patients is that when you wear a mask, what you're really doing is you're preventing your respiratory droplets from getting into the environment. So you're actually protecting other people. Unfortunately, if somebody with COVID-19 sneezes in your hair, you're probably going to get infected because you're going to touch your hair at some point that is going to eventually get into your nose and mouth, et cetera. At some point, you're not wearing a mask. So masks actually are important public health measure because they prevent people who may be unwittingly or knowingly exposed to the virus from causing other people to be infected by putting their droplets out into the air. But then you see people that get so crazy about it. I see people that are in a parking lot and they're so worried that their mask is fitting perfectly in the parking lot and then they're not even close to anybody. And it's nice that they're concerned about it, and I like the focus. But the reality is that you're not going to pick up COVID-19 in the middle of a parking lot based on breathing the air. So I think people sometimes have misconceptions about what the masks will do and what they won't do. Social distancing is important. My favorite example of that was going to a restaurant 3 weekends ago, and I have definitely taken advantage of the fact that the restaurants here in Florida have opened. I've gone to places that are either open air or have practiced social distancing in terms of the table setup, and I've been even asked by wait staff if they should wear a mask while they serve me, and I said, absolutely, you should wear a mask while you serve me. That's, I think, a very important gesture, and who knows, maybe you have an asymptomatic infection, and I would appreciate you wearing your mask. But I think the risk in that setting for the patrons at a restaurant is really quite small. On the other hand, I was at this restaurant that was doing a good job of social distancing in the eatery area, but next door there was a bar, and you can see the bar scene through a glass window. And I looked at the bar scene, and they were playing music, and people were slobbering over each other, and they were drinking out of each other's glasses. And I was saying, this is a disaster. And this is 3 weeks ago, before we had this surge of cases in Florida. So it was no surprise to me why that occurred. So again, I hope people use common sense and understand that you can still enjoy life. You can still go and get into your car, and you can go to a store and just stay your 4 to 6 feet away from everybody. Wear your mask when you go get your groceries, when you go get your your dry cleaning, etc. And you're safe. But don't do silly things like go to bars with a bunch of people. Stay out of big groups. If you want friends coming over your house, it's okay. Bring them outdoors. If you have a patio, you serve them drinks outside, and just stay 6 feet apart. It's very doable. We had a nice little party this weekend with 10 people, no more than that, and we had set up the chairs where everybody can sit apart from each other, and we had wipes, and it was all good. And I'm pretty sure we didn't infect anybody there. So again, I think my message is, yes, we can live our lives and enjoy ourselves by taking some sensible common sense precautions. Where should I refer patients to get into a trial? So our website would be www.encoredocs.com or www.jacksresearch.com, but the new website is Encore Docs, but you'll be directed there from our older website. And just let us know that you have an interested patient and give us contact information, and we'll reach out to them. We also have a phone number, 904-730-0166. And that's our phone line, and we'll have one of our recruiting staff take the information and reach out to the patient. We're very excited about the program. I personally have already referred a number of my own personal patients, and people have run into the hospital. I think healthcare workers are actually a good target for this program for a number of reasons. One, they understand the process perhaps a little bit better than others, and two, they're probably at a little bit higher risk for infection. So we would encourage those people. Other people that have a particular concern about being at high risk, waitresses, waiters that work at restaurants that have to interact with the public may be good candidates. So just think through who would be good candidates. People who would not be good candidates are people that have pre-existing autoimmune conditions. If you're on chronic steroids, you wouldn't be a good candidate, of course. And of course, we'll go through all that screening when patients come to our center. But as a physician community, I think it's important for us to reach out to a lot of patients, let them know that we're really excited here in Florida to have access to this vaccine through research, of course, before a lot of other communities. And there are certainly a number of patients, particularly in our surge condition right now, who may benefit through this experience, if nothing more, by learning a lot more about how to protect themselves against COVID-19. Any other questions? Well, I will wrap it up then. Thank you for your attention, and we certainly live in interesting times. We are in the midst of a crisis, but I like to remind my staff that the Chinese symbol for crisis contains two other symbols. One is danger, and the other is opportunity. So I'd like to look at this crisis as our opportunity to use a medical solution to make the lives of our patients better, and hopefully to protect patient populations in the future, and using the learning to not only protect people from the current disease, but future illnesses. And I thank you for your attention. Have a great day and be safe.